Acid-base balance. The medium that sustains biochemical, cellular activity, movement of ions and literally key functions of enzymes and living body. In short, the medium that sustains life. As a doctor, it is essential to know the basics of acid-base balance so that well thought out treatment is extended to the patients. As we go through this series of ABGs, we are going to first get a hold of basics of biochemistry, physiology from the eye of traditional approach. In episode 2, we will dive into compensatory mechanisms, and in episode 3, we will practically read the ABGs with learned mind. In episode 4, I will introduce you to steward approach, which is a more advanced approach to viewing the pathophysiologies of acid-base balance. And in the final episode, we will dive into the treatment and management once the diagnosis is made through good ABG reading. Let's get started then. So this is how an ABG strip looks. We will focus on 4 entities here. Note the normal indices here. pH of 7.35 to 7.45, PCO2 of 35 to 45 millimeters of mercury, bicarb at 22 to 28 milliequivalents per liter, and base excess or base deficit resting at -2 to +2. Note PO2 and its relation to fraction of oxygen FiO2 being given to the patient at the time sample was taken. This is about the AA gradient and PF ratios, something we will target through our respiratory system, playlist and oxygen cascade system. Hydrogen ions control the pH of solutions. Traditional approach relies on carbon dioxide and bicarbonate in confirming the net hydrogen ion concentrations. In body, as per Henderson Hasselbalch Equation, the concentration of free hydrogen ions can be calculated as follows. Adding normal values of CO2 and bicarb ion, it turns out to be 40 nano equivalent per liter. Since hydrogen ion is in nano equivalent compared to milli equivalents of bicarb, the nano equivalent is such a short number it is expressed in negative logarithms. So dare I say that hydrogen ion concentration is inversely proportional to pH. So on pH scale, relating to the normal values of ABG strip, 7.35 to 7.45 is green or normal zone. In terms of hydrogen ion concentration, 40 nano equivalents is 7.4 pH. So hydrogen ion concentration between 32 to 50 falls within green zone or physiological normal pH. Hydrogen ion concentration between 160 nano equivalent to 60 nano equivalent is compatible with life, that is pH of 6.8 to 7.8. Beyond this range, life is impossible. So I have marked these maximum zones in yellow. So let's place this equation visible for future reference as 1.1. Also note the traditional approach is relying heavily around CO2 levels and bicarb levels to dictate pH calculations. The definition in traditional approach is based on Bronsted-Lowry elaborations that acid is any compound that gives off or donates hydrogen ion, whereas base is any compound that receives or accepts hydrogen ion. Now, throughout this lecture, I will mark red for acidity, green for neutral, and blue for alkaline. Suppose we have a flask full of green colored neutral pH fluid. So let's add an acid to it. As per the definition, an acid donates hydrogen ion. So HCl dissociates in water and gives off hydrogen ion. This would increase the net hydrogen ion concentration in fluid and make it acidic by lowering its pH. Since we just said hydrogen ion concentration is inversely proportional to pH. Any acid that completely dissociates into ionic form in water is called strong acid. Carbonic acid partially dissociates, so it exists as unionized compound and its ionic dissociation molecules. This is thus a weak acid. Now let's see how base reacts in neutral water. So bicarb has a negative valency, it attracts hydrogen ions and forms compound. In other words, free hydrogen ion concentration is lowered. So pH of the solution would increase. So it is hydrogen acceptor. Next in definition comes the buffer system, which keeps the pH within neutral range on addition of acid or base to the solution. For example, the bicarb buffer system or the carbonic acid. We said carbonic acid is weak acid that is partially dissociated in water. So now when you add hydrochloric acid, as soon as hydrogen from HCl enters fluid containing bicarb buffer system, the menacing hydrogen ion is immediately taken up by bicarb and converted in a series of reactions to carbonic acid and carbon dioxide. The carbon dioxide is then easily washed away through lungs, so the pH remains stable. Similarly, if we add sodium hydroxide as a base on dissociation, it is trying to raise the pH of solution, but hydrogen ion of buffer system will now react and form neutral H2O. In this way, it will buffer it up. So what determines whether carbonic acid buffer exists more as ionized form or as unionized compound? It's the pH of the solution which directs it. The pKa is that pH where ionized and unionized forms of weak acid or weak base exist at 1:1. To understand the pKa, let's take local anesthetics for example. To be effective, local anesthetic must first be in unionized form so it can penetrate through tissues easily through diffusion. And once inside the cell, it is the ionic form that blocks the sodium channels. So initially you need local anesthetic to be in unionized form. The pKa at which ionic and unionic forms exist at 1:1 for local anesthetics is 7.8 to 8.2. So when local anesthetic is injected into abscess for abscess drainage, the abscess area has acidic pH. So local anesthetic will be more in ionized form than unionized form. So it will not diffuse around easily and its effect will be badly compromised in reducing pain. So clinicians add soda bicarb to local anesthetics to theoretically increase the pH of tissue so that pH gets nearer to pKa value of local anesthetic in an effort to have more unionized form available for better diffusion and better effect of local anesthesia. So now you know significance of pKa values. Below 7.35 pH, there is either too much acid in the body or too less base. This is called acidosis. The opposite occurs in alkalosis above pH of 7.45. We said traditional approach at looking at acid-base balance revolves around the carbon dioxide and bicarb levels and Henderson Hasselbalch equation. Note, as per this equation, if you increase the base in body, pH increases. And if you add acid in denominator of equation, the pH would fall, right? So in carbonic acid buffer system, CO2 is acid and bicarb on the right side is base. The pKa of this carbonic acid system is 6.1. So as per equation now, 6.1 plus log of bicarb as base and carbonic acid as acid. So we said pKa is the pH where ionic and unionized forms exist at 50:50. So in this scenario, keeping bicarb base and carbonic acid at 1:1, the pH would turn out to be 6.1. This means that if body's pH is lower to 6.1, it would mean a 1:1 proportion of carbonic buffer, right? So to keep pH of body at physiological ranges of 7.35 to 7.45, we technically need to increase the base, right? See the equation. Compared to acid. This is represented in ABGs where base is 22 to 28 milliequivalents per liter. Now solving the equation. 24 as normal level of bicarb, 1.2 for carbonic acid. So the pH of solution becomes 7.4, which is normal pH. What is this showing you? This is showing that to keep blood or extracellular fluid pH at 7.4, the bicarb is 20 times more than its acidic counterpart. This 20 ratio is reflected in ABGs as 22 to 28 milliequivalents per liter range of normal bicarb. This is called the alkaline reserve. Add acid and bicarb gets consumed to neutralize hydrogen of acids. When bicarb decreases, it means the ratio is shifted now from 20:1 to more towards 1:1 side or more towards acidic 6.1 pH. This is reflected through Henderson Hasselbalch equation. Lower the bicarb and so you are lowering the pH. So far, we have seen how carbon dioxide and bicarb make a fine balance to maintain the pH at 7.35 to 7.45. Carbon dioxide is under respiratory control, whereas bicarb is under metabolic or renal control. What if on this seesaw scale, we shift the weight towards acidic environment, either by increasing carbon dioxide or reducing bicarb? Then pH would decrease, and below 7.35 it is called acidosis. If due to increase in carbon dioxide, it is termed respiratory, and if due to reduction in bicarb, it is called metabolic. Not a rocket science there, right? On the contrary, if we reduce the carbon dioxide in denominator or increase the bicarb, pH would move upwards. This condition of pH greater than 7.45 is called alkalosis. If due to reduced carbon dioxide levels, it is called respiratory alkalosis, and if due to raised bicarb, it is called metabolic alkalosis. Remember the mnemonic for detection of primary anomaly. For respiratory or carbon dioxide, the pH and CO2 move in reverse manner. So the mnemonic reverse, where the RE in reverse stands for respiratory. So if carbon dioxide moves up, pH goes down. If carbon dioxide goes down, pH goes up. For metabolic disorder detection, pH and bicarb go in same direction. So the Me at the end of same is for the Me of metabolic. Bicarb down, pH down. Bicarb up, pH up. So same direction. Remember these two mnemonics. We will practically apply them on different ABGs in episode 3. There are three main defense systems in the body to maintain pH in normal range. Number 1 is of course the buffer system. Number 2, the respiratory, that is lungs, controlling carbon dioxide washout. And number 3, the renal system for bicarb regulation. Now all these systems are interlinked. This I'll explain as we move through individual systems. So let's see the buffer system first. The main buffer system is the bicarbonate or carbonic acid buffer system. We have seen how its pKa is 6.1, so it is most effective around 1 pH above or below the 6.1 mark, meaning it should be given when pH in body is around 7.1 or below, right? The second important buffer is the phosphate buffer system. Its pKa is 6.8, which is closest to physiological pH of 7.4. So it exists almost in 1:1 ionic, non-ionic form. Hence, it is actually the most efficient system, more than bicarb in fact. At physiological pH, the bicarbonate system has 20:1 in favor of ionic bicarb, whereas phosphate exists in 4:1. Then why do we still target bicarbonate buffer as key to maintenance of pH? Firstly, because of its immense quantity compared to other buffers. Secondly, it is under physiological control. The carbon dioxide and bicarb components are under control of lungs and kidneys. So bicarb buffer is most important in extracellular space and phosphate buffer in intracellular space. The third buffer worth mentioning is albumin, proteins and hemoglobin. They will be mentioned as we move on in this episode. Let's see how these defense mechanisms are connected. Peripheral tissue metabolism consumes oxygen, gives off carbon dioxide as a byproduct. The hemoglobin in periphery unloads oxygen because under acidic environment, hemoglobin oxygen dissociation curve shifts rightwards. This is also called the Bohr effect. This frees hemoglobin, which then acts as buffer of hydrogen ions. How? Let's see. So the unloaded oxygen molecules enter peripheral tissue while carbon dioxide comes out into extracellular fluid, where it either exists as dissolved form or enters red blood cells, where two things happen. The free hemoglobin attaches carbon dioxide, forming carbaminohemoglobin, or more importantly, carbon dioxide gets hydrated and forms bicarb and free hydrogen ion. This free hydrogen ion is then taken up by the hemoglobin buffer system, whereas the bicarb moves out of the cell in exchange for chloride. This is also called the chloride shift. So we see the venous system has more carbon dioxide content than arterial system. Next, the red cells travel through the venous system into the lungs, where alveoli supply oxygen, and under Haldane effect, The hemoglobin has more affinity for oxygen than carbon dioxide, so it unloads carbon dioxide, takes up the oxygen. The freed up carbon dioxide diffuses easily through the red blood cells, through the alveolar capillary basement membrane into the alveoli and out in exhalation. The process is reversed for hemoglobin buffering the hydrogen ion. Bicarbonate enters the red blood cells and combines to hydrogen ion, generating carbonic acid, which further forms carbon dioxide. The carbon dioxide similarly is evacuated through alveoli and out of body. So now you see why carbon dioxide is acidic. And in Henderson Hasselbalch equation, carbon dioxide is taken as acid part of denominator, because this carbon dioxide is linked to the generation of hydrogen ions once carbon dioxide gets hydrated with water molecules. And in the lungs, the opposite occurs, where free hydrogen generates carbon dioxide that is then washed out. So on the seesaw scale, we can safely write hydrogen in brackets with carbon dioxide, right? Now remember this equation. The amount of carbon dioxide in body is determined by carbon dioxide production divided by alveolar ventilation. So when carbon dioxide increases for any reason, it generates more hydrogen and lowers the pH. This lower pH is then detected by chemoreceptors in periphery and in brainstem medullary centers. So the brain immediately increases the minute ventilation by 1 to 4 liters per minute for every single rise of carbon dioxide above 40. So increased ventilation as denominator would then cause decrease in carbon dioxide and stabilize the pH. So in this way, hypoventilation or hyperventilation directs carbon dioxide content in blood and hence directly gets involved as defense against changing pH. Coming to the kidneys' role, kidneys control acid-base balance in 4 ways. The first 2 processes marked in pink occur in proximal convoluted tubule, the hydrogen ion secretion and the bicarb reabsorption. The next 2 processes occur in distal convoluted tubule marked in orange, namely the titrable acids and the ammonium buffer system. Let's dive into the proximal tubule. Now remember, urine is being made in tubular lumen, right? So in the cells of proximal tubule, the carbon dioxide hydrates under carbonic anhydrase enzyme, forming carbonic acid that further dissociates into hydrogen ion and bicarb ion. This hydrogen ion is exchanged for sodium ions and is transported to tubular lumen, where it can either be secreted in urine or it could be utilized to return the filtered bicarb ions that ran into the tubule from glomeruli. Combined together, same process occurs and carbon dioxide is generated, which diffuses easily into the cell, and the whole process repeats under carbonic anhydrase enzyme. So you see how bicarb goes through a series of steps to be transported from tubular lumen to cell and out into the bloodstream again. This is called the bicarb reabsorption. Note, this is how carbonic anhydrase enzyme inhibitors like acetazolamide causes acidosis. By stopping the enzyme action, it causes decrease in hydrogen excretion and bicarb reabsorption, thus increasing the acids and reducing the base. In the distal tubule, disodium phosphate gets ionized into sodium and monosodium phosphate. This sodium is utilized in bringing the hydrogen ion into tubular lumen, which then forms compound that is excreted in urine. So does that mean sodium has a role in guiding acid-base balance? We will see that in concept changing mindset when we start more advanced talk, that is the Stewart approach. The next important thing is of course the ammonium buffer system, which helps in excretion of hydrogen ions. So you see how these 3 defense systems are interlinked, all working in harmony to control the carbon dioxide and bicarb ratios that further direct the pH of body, adding to the set of equations the CO2 content in the body. Let's just stop here for now. In the next episode, we are going to look into the compensatory mechanisms, the 6 formulas that need to be crammed up in order to understand the partial compensation, the full compensation on ABGs. There are other vital concepts such as anion gap and base excess. We're going to discuss that next. Stay tuned.